Waste is a part of life, and many systems of the body have to deal with waste. So the respiratory system has to exhale waste carbon dioxide. The digestive system needs to eliminate things which can't be digested. Sweat glands can eliminate waste, etc. One of the main ones, however, is the urinary system. This is an amino acid. These are the building blocks of proteins like the ones that you eat in your diet. Amino acids get their name because each amino acid has an amino group, that's the NH2 group here, and the carboxylic acid group, the C double bond O, OH, and therefore they're called amino acids. If you want to use an amino acid for energy, you have to remove the amino group, the NH2. But then the problem is that this could form ammonia, NH3, which is highly toxic. The liver helps out by converting two potential molecules of ammonia into urea, which is less toxic, but nevertheless, it needs to be eliminated from the body. If your kidneys were to fail, then that's extremely serious, and you might now have only hours to live. Now, I don't want to say that the only purpose of the urinary system is to limit, to excrete urea. It controls the volume and content of body fluids, it secretes hormones. There are other functions, but releasing urea is certainly one of its primary functions. This is a smaller system. It's composed of 2 kidneys, 2 ureters, 1 bladder, 1 urethra, and that's it. The kidneys are referred to as being retroperitoneal. So while the digestive organs are in the peritoneal cavity, the kidneys are behind that in the, so they're called retroperitoneal. So you'll notice here that while the intestines are free to move in the abdominopelvic cavity, that the kidneys of this cat are behind this. All right. They can be held in place by adipose. One of the potential consequences of losing too much weight, you can see the adipose around this overweight cat's kidney here. One of the consequences from losing too much weight might be that the kidney could dislodge. The kidneys receive an incredible amount of blood, certainly a fifth and maybe even up to a quarter of the blood which leaves the left ventricle at rest heads towards the kidneys. The abdominal aorta produces renal arteries, which then form a number of branches before the veins return back, forming renal veins. And so the body certainly takes the elimination of urea quite seriously. Once again, over a fifth, maybe even almost a quarter of the blood leaving your left ventricle can be sent to the kidneys for urea to be filtered out of the blood. We can split the kidneys into an outer cortex and an inner medulla. The inner medulla is composed of pyramids and renal columns then between them. And it is here that urine is made. Once urine is made, it drains into small spaces known as minor calyces. Minor calyces fuse to form larger spaces known as major calyces. The major calyces empty into a central portion known as the renal pelvis. And from here, a urine can pass into the next section, the ureter. Now, although we could get into the microscopic aspect of the kidney at this point, let's just mention all of the macroscopic anatomy first, and then get back to the microscopic anatomy of the kidney, which is the majority of the chapter on the urinary system. Okay. So from the kidneys, urine is made, and then it simply needs to be released. So it can then empty into the ureters, which could be 10 inches, a foot long, and the ureters pass urine both through gravity, but also through peristalsis, the muscle contractions which pass over the ureters through their muscularis layer in waves. The ureters empty into the one single bladder and then can be released through the one single urethra. The ureters empty into the bladder obliquely. They come in from an angle, and this way, as the bladder fills, it puts pressure on the ureters to prevent a back flow of urine. The bladder is composed of transitional epithelia, but so too is the distal portion of the urethra. My fault. The ureter. Moving into the bladder. So once again, all of these videos are here. They go into this in a bit more detail. The bladder is lined by transitional epithelia, which can stretch. It has that name because one can't call the cells cuboidal, which is how they look when they are relaxed, nor squamous, which is how they look when the bladder is full. They first evolved in female cartilaginous fish. That was why that image was there. And so the transitional epithelia can't be named because the cell, by the shape of the cells, because this cell then changes. Transitional epithelial cells can then be found in normal urine, and this is not unexpected, given that epithelial cells can slough off the surface of the epithelia into urine, and so their presence is to be expected. There is smooth muscle in the wall of the bladder that is subject to involuntary reflexes, and one could imagine that you could draw an imaginary triangle between the openings of the two ureters and the exit of the one urethra. So since there are three things which leave the bladder, two ureters come in, one urethra leaves, one could draw an imaginary triangle known as the trigone. The one single urethra, which exits from the bladder, is lined by stratified squamous epithelia, and thus as urine then passes, squamous epithelial cells can then end up in, it starts off as transitional near the bladder, but then here you can see those flat squamous cells there, and that's important because once again, if one was doing urine analysis, there are things which are normal components of urine, and so squamous epithelial cells from the urethra and transitional epithelial cells from the bladder are things that can normally be associated with urine. Another point worth mentioning is that in half of female mammals, there is some fusion of the urinary system and the reproductive system, but that doesn't apply to primates, including humans. And so the urethra in females is only a urinary structure. It empties separately into the vestibule from the vagina, and so therefore the urethra is urinary only. In contrast to the urethra in males, which passes both urine and semen, and as such is part of both the urinary and reproductive systems. Its increased length in males then has one other consequence of then increasing the distance between the exit of the urinary system and the urethra and the exit of the digestive system, which makes males less susceptible to urinary tract infections, given that most bacteria which cause urinary tract infections came from the digestive tract. So this is the macroscopic anatomy of the urinary system. But much of the chapter is dedicated to the microscopic anatomy of the kidneys. Now, when studying muscles, it's commonly said that the sarcomere is the functional unit of muscle. If you can understand how sarcomeres work, then you can understand how muscles work. The alveolus is the functional unit of lungs. In the same way, the nephron is the functional unit of kidneys. If you can understand how nephrons work, you understand how kidneys work, since a kidney is about 1 million nephrons. Nephrons have two parts. They have the renal corpuscle, which is essentially capillaries in a bag. This is where filtrate is made. And then from this renal corpuscle, one enters the renal tubule, which can be divided into sections. And so nephrons, the functional units of kidneys, have two parts, the renal corpuscle and the renal tubule. Let's begin with the renal corpuscle. Kidneys are odd in that in the pathway of blood flow through the kidney, at one point, an afferent arteriole forms glomerular capillaries, which are then drained by an efferent arteriole. Nowhere else is a capillary bed drained by arterioles, only in the kidneys. Why this unique circulatory setup? Well, the reason is that these capillaries are special. Unlike other capillaries, which are trying to service a tissue, provide cells with glucose and oxygen and the like, these capillaries are here for one reason only: to leak. There's poison in the blood. This urea will be toxic if it builds up to enough quantities, and so it has to come out. And so that's why these capillaries are here, to leak. There needs to be less urea in the efferent arteriole leaving the glomerular capillaries than there was in the afferent arteriole, which formed these capillaries. And because we want this urea to leak out, this glomerular capillary set of capillaries is surrounded by a bag, a glomerular capsule formed by simple squamous epithelia. And this encloses a space, a capsular space. So when stuff leaks out of these glomerular capillaries called filtrate, it will go from the capsular space into the beginning of the renal tubule, which is known as the proximal convoluted tubule. Now, when I said capillaries leak, in part that's obvious because all capillaries leak at least a little bit. I mean, gases can come out, and most capillaries have spaces, intercellular clefts, which at least allow some stuff to come out. But the capillaries in kidneys are fenestrated capillaries. They have pores known as fenestrations, which are meant to be extra porous. And so not only do all capillaries leak at least a bit, these capillaries, they leak to a greater degree than is typical because of the pores, the fenestrations. So stuff is going to come out of the blood here and then leak into that capsular space. But to do so, anything that comes out needs to fit through two spaces. First, this space here called a fenestration. This is a fenestrated capillary, so this then is a fenestration. But also on top of the fenestrated capillaries are these foot cells or podocytes. And podocytes have these processes called pedicels, and there are filtration slits where two pedicels meet. So in order for something to leave the blood and then potentially enter the filtrate and potentially the urine, it has to be able to fit through two things, through the fenestration and then through the filtration slit. Now, blood cells are too big, and so therefore blood cells should not be in urine. Proteins are too big, and therefore proteins should not be in urine. When we cover urinalysis, urinalysis, we'll go through what would cause different substances to be present in urine when they shouldn't. But smaller things can fit through these. So water can obviously pass through, and it can bring small molecules like glucose, amino acids, salts. All of these are good things which need to come back and be reabsorbed at some point. Sorry. But then also urea can leave, which this is the main purpose for having kidneys, is to extract urea from the blood. Now, I had said that the nephrons can be divided into the renal corpuscle and the renal tubule, and one can see this under the microscope. So the renal corpuscle is capillaries in a bag, and you can very clearly see here are the capillaries surrounded by a capsular space, surrounded by the simple squamous epithelia forming the glomerular capsule. So you can see in a cross-section of the kidney all of the renal corpuscles. And then you can also see the renal tubules. Most sections of the renal tubule are lined by simple cuboidal epithelia, although the descending loop of Henle is lined by simple squamous epithelia. And here you can see here, this is clearly the renal tubule. You can see the lumen in the center and these cuboidal cells alongside. So you should be able to look under a microscope and then distinguish between the renal corpuscle and then the renal tubules, which you see here. And now, okay. I think I missed one section here. So just pointing out that these are, once again, fenestrated capillaries, so stuff is going to come out. It's what's called filtrate. It's not urine yet, and here's an obvious way to show that. The average person may say 1 liter or 2 of urine in a day, but a kidney can make 180 liters of urine, of filtrate in a day. So if filtrate was the same thing as urine, there's a big difference between 180 liters and 1 to 2, that, obviously we would spend much more time in the restroom. And so there are fenestrations, holes in the capillaries. These are fenestrated capillaries. But then there are these foot cells, these podocytes, whose processes, pedicels, contact each other. And so anything that leaves the blood and enters the filtrate has to then move through 2 things. One, the fenestrations, but then second, the filtration slits between these foot cells. So this is what then becomes the filtrate. And once again, big stuff can't leave: blood cells, blood proteins. And so the filtrate then has water and things small enough to pass with it, like glucose, amino acids, salts, and urea. The renal tubule can be split into 4 sections. The first portion is very bendy and it's close to the renal corpuscle, so it is called the proximal convoluted tubule. Proximal because it's near the renal corpuscle, convoluted because it winds. So the proximal convoluted tubule, or PCT, is the first section. Then there is this loop of Henle that goes down and then up. There is a descending loop of Henle, followed by an ascending loop of Henle. There is a distal convoluted tubule. And then this is where the nephron officially ends. Multiple nephrons then empty into a collecting duct. This empties into the papillary duct, which empties into the minor calyx. And you might remember the minor calyx from a previous video. So anatomically speaking, in addition to the macroscopic gross anatomy, there are features of the microscopic anatomy of, wrong one, of the microscopic anatomy of the nephron. The last kind of, as we wind down on the anatomy of it, one last, major point is that the end of nephrons, all right, so if the renal tubule is essentially just a tube, the tube can go in any which way. It could go this way, it could go that way. But it actually, so this tube, where's it going? It could go anywhere. But you know where it ends up? It ends up right here, all right? The end of the nephron comes back to touch the beginning of the nephron, all right? And so where it does so, this is a special region called the JGA, the juxtaglomerular apparatus. The end of the renal tubule then doubles back to contact the beginning of the nephron at the afferent arteriole of the renal corpuscle. And so here there are two special kinds of cells. There are toll cells of the distal convoluted tubule called the macula densa. They are important because they sample the amount of salt in the filtrate and thus determine whether or not you're making the appropriate amount of urine. And then there are specialized smooth muscle cells over the afferent arteriole called JG cells or granular cells that make the hormone renin. And so where the end of the nephron touches the beginning of the nephron, there's a very important regulatory aspect with these two cells, the macula densa, the toll cells of the DCT, and the JG cells over the afferent arteriole making renin. Two final really quick points. Nephrons can vary in how long their loop of Henle is. The loop of Henle can be short, and so thus then the entire nephron can more or less be in the cortex. So all of the renal corpuscles are in the cortex of the kidney. And if you have a short loop of Henle, then the loop of Henle is as well. So those are called cortical nephrons. And then the, while other nephrons have very long loops of Henle, and they can then dip down into the medulla, and these then are known as juxtamedullary nephrons because of their long loops of Henle. One final just anatomical term. So the afferent arteriole forms the glomerular capillaries, which then forms the efferent arteriole. This then will, will form, so here comes the afferent arteriole coming in. It forms that renal corpuscle. It'll then form the efferent arteriole, but then it forms capillaries. What do we call the capillaries? Well, there are two names. If the capillaries surround the PCT and DCT, then they're called the peritubular capillaries. If, however, the capillaries surround the loop of Henle, it is called the vasa recta, all right? Both the peritubular capillaries and the vasa recta, they empty into the venule, which will then empty into the veins and return fluid back to circulation. So this then is the anatomy of the urinary system.